Incoming call.
uh, mostly cancer cells, by altering the receptors on the surface of the T cell so that they can go after kinds of cells that they can't naturally go after. Well, uh, CAR-NK is using the same kind of technology but applying it to a different kind of cell called NK cells. And the advantage here is that NK cells are already the kind of cell that our body uses to attack senescent cells already. And so when you develop CAR NK cells, you are both introducing new receptors that can go after senescent cells in new ways, but still retaining the existing inbuilt capacity of NK cells to destroy senescent cells. And so he'll be talking a bit about that. Um, he will also be talking about ways that he has discovered in the process of looking for new receptors that he could use to weaponize NK cells using this CAR NK approach. Um, he also has discovered peptides that have inherent senolytic activity, and so how he could use that, and also a completely new sort of classic small molecule senolytic approach using a new vulnerability in senescent cells that he discovered in the process of doing unrelated work. Dr. Sharma, I would have to say, is very, very creative and observant. When he makes a discovery, he sees better than a lot of scientists can how this can be turned into a therapeutic approach. And so, just like a happenstance discovery in the process of research, he drilled down into it and discovered a way that is looking very promising using small molecules to destroy senescent cells on the basis of that discovery. Um, and also, it's interesting because when you look at different studies of how the immune system surveys senescent cells, in different contexts, different cells seem to mediate most of the work. And so there's one set of cells that has been shown in models of tumors after senescence has been induced by chemotherapy, in a model of liver disease where you get hepatocytes that have gone precancerous. I can hear someone who is talking to me. Yes. Huh. I'm going to continue with my talk, even though someone is apparently trying to talk to me. Um, uh, so, Dr. Dr. Sharma, this paper back in 2012 predicted that there would be a kind of immune cell that would be responsible for cells that accumulate as a result of aging, as opposed to these other processes. And I think Dr. Sharma may actually have discovered it. I'm going to briefly pause because I think someone is trying to speak with me now. Huh. Pardon me? I'm welcome to continue. Okay. Sorry. Bit of confusion there. Um, okay. So the next area that we're going to be highlighting today is mitosens. So this is the problem that mitochondria, although they cause a bunch of different functional changes with age, most of those are secondary to other kinds of problems elsewhere in the body. The one that is most clearly directly happening in the mitochondria themselves is the accumulation of mutations, and in particular, these large deletion mutations. Um, I'm delighted to say that Dr. Blumenautham, along with colleagues here at SENS Research Foundation, after a long period of stagnation in the field, has been making really excellent progress in the last few years. Uh, Back here, this was several years ago, both showing for the first time that allotopic expression not only could restore the function of uh, the mitochondria that had accumulated mutations in this mitochondrial genes ATP8 and ATP6, but really robustly showing that allotopic expression was actually doing that by restoring oxidative phosphorylation, which previous reports on allotopic expression have never clearly shown. This was the clearest and most decisive thing, and it really eliminated the skepticism in terms of is allotopic expression really working. She's going to talk a bit about her subsequent progress there, in particular on two genes, ND4 and COX2. Um, in addition to that, uh, up until now, all of this progress has been happening just in cells, in isolation. And of course, we want to work it eventually in living, breathing humans. Well, we have gotten the next best thing. Dr. Bhuvanathan has actually achieved this in living mice. So these are mice with a mild mutation in ATP8 through a combination of a technology that we developed thanks to Dr. DeGray's foresight and her allotopic expression work. She can now put the allotopic version of ATP8 into the cells of mice and show that it restores their mitochondrial function as we would expect from allotopic expression. Um, she also has another approach she's just going to touch on briefly. Um, Mitochondrial large deletion mutations occur because of a change in the way that the cell carries out quality control. So a lot of you will have heard of mitophagy, which is the process whereby cells recycle their mitochondria, eliminate abnormal mitochondria, and replace them with replication of the healthy mitochondria elsewhere in the cell. Well, it turns out that in the case of mitochondria bearing large deletions, this whole mitophagy machinery fails. And so they fail to see, it appears, the mutation bearing mitochondria, and they just accumulate and eventually completely take over the cell. And this obviously is a major problem. Um, in this new approach that Dr. Manathan will tell you about, she is working on a way to essentially eliminate that invisibility shield that these guys have and make them once again susceptible to mitochondrial turnover, mitophagy, and being replaced. And so that could not restore a cell that has been completely taken over by mutation bearing mitochondria back to normal because once it's 100% mutation bearing mitochondria, there is no wild type mitochondria to repopulate with, but rather if you do this early on, you could forestall the date at which the cell becomes taken over and thereby extend the normal activity of that cell and bias time, basically. Uh, and Dr. Riba has been working for some time in iron metabolism and aging. That actually uh, began before he came to SENS Research Foundation to some degree. Um, and he is now working on that further, looking at iron in a variety of contexts, and in particular in heme iron, the kind of iron that carries oxygen in our cells that we normally think of as being a safe storage form, but under some circumstances can become quite pathological. And this also builds on some work that was previously done at the SENS Research Foundation Research Center by Dr. Tesfahun Admatsu, who was our postdoctoral fellow, who discovered both the resistance of a whole class of senescent cells to most of the mainstream senolytic drugs, and also a new iron-based strategy to eliminate senescent cells. Um, I am getting dangerously close to the end of my time, so I think I will stop, unfortunately. But there is also exciting work happening extramurally with Dr. Jean Hebert, repopulating the neocortex, um, and a project that I am very excited with that it took us uh, literally more time than it would take a baby to get going with Dr. Annalise Barron at Stanford to develop new analogs of a protein in our body called LL37 that will have dual effects against Alzheimer's disease. And finally, some work on the biology of lithium in the brain by Dr. Evan Snyder, who is just fabulous in terms of training many of the young rejuvenation researchers that we put out through SRF education program. I apologize to all three of you extramural researchers for not giving you your full due, but I'm very excited by everything that all three of you are doing, and will look forward, as everyone should, to your presentations today.
Okay, everybody, I think we're having a little bit of a technical difficulty. We will be right back with you. Don't go anywhere. We have great stuff coming. Okay, sorry about that. A little bit of trouble with technology. We need more AI, I guess. So we are going to have Dr. Amit Sharma on stage now. He leads our Apoptosense program at our research center in Mountain View. He's, as Michael Ray just talked about him, at our part of our intramural research. His presentation today is next generation xenotherapeutics, from harnessing the immune system to targeting newer vulnerabilities of senescent cells. You can read his presentation summary and bio on the events website. And I think Amit is about to be ready, and I can welcome him on stage. Yes, there he is. Thank you, Amit. Thank you so much for the amazing work that you do for us. We really, really appreciate you. And can't wait to hear your presentation. I think we cannot hear him. Yeah, we can hear him. Okay, awesome.
Hi,
Yay.
Maria.
I'm so excited. Oh, and I want to say thank you to Michael Ray also for a wonderful presentation. That was awesome in explaining about the fact that we're not trying to slow down aging, but to actually fix this horrible medical problem. So go ahead, Amit.
All right, I'm ready. Thank you, Maria, and thank you, Michael, for that amazing introduction. I am going to talk about some of the work that has happened in the lab, where we are trying to find ways to eliminate senescent cells. Michael kind of stole a little bit of what I was going to talk about Sense Foundation and why we are in existence. So it may be preaching to the choir, but we are increasingly becoming old. So this is the data from the latest Census Bureau. In 2022, by 2020, the total proportion of people over the age of 65 in this country has gone up significantly. With that, there are people with many age-related diseases have also gone up. So a better understanding of how to target those, those diseases and how to target aging really can help us understand, develop therapeutic interventions for this, and actually improve the quality of life and improve our society. So, what is aging? I mean, on surface, this may be a very simple definition. This is what we were considering aging as an inevitable consequence of growing. So the definition that people would normally follow would be the deteriorative changes with time, post maturation life that underline increasing vulnerability to challenges, thereby decreasing organisms' ability to survive. That was the definition of aging, and every scientist who was studying this would consider that as an inevitability. The foundation of Sense is based on one of the first papers that was published from Sense Foundation, which talked about these collective accumulation of these changes that accumulate over lifespan of a person or an organism, which can cause molecular and cellular, which cause changes in molecular and cellular structure of an organism that mess with the metabolic processes, which then contribute to increasing age and increased pathology, and eventual death. With that idea in mind, and being able to target those changes that are occurring with increasing age, Sense came into existence. Our basic idea is to not only do research. So Sense Research Foundation is basically doing intramural and extramural research on aging, but we are also involved in advocacy and educating next generation of scientists. Like Michael was talking about, aging is a result of all of the accumulation of damage that occurs during the lifespan of a person. What are those damages? We know there is increasing evidence of accumulation of intracellular and extracellular protein aggregates. Amutha will talk about this a little bit later. The main energy source in our body, mitochondria, during the process of age can undergo lots of mutations. Those accumulated mutations can also contribute to inflammation and other kinds of damage. We know our chromosomes become shorter, the telomeres become short, and they start accumulating DNA damage. Many of these damaging events can lead to accumulation of senescent cells in our body, and I'll talk about senescent cells in a little bit. These senescent cells produce a lot of pro-inflammatory cytokines, chemokines, and enzymes that modify our matrix, make it very, very stiff, and disrupting organ tissue function. And with this inflammatory environment, our ability to repair damage diminishes quite significantly, and we experience a stem cell exhaustion. Like I was talking about, a combination of all or many of those damages can lead to aging. And if we look at this as an engineering problem, then this can be solved. We can target those individual damages, and then we can maybe target aging. And one damage I didn't mention is the immune senescence, and the immune function loses its ability to function properly, and our ability to repair damage also declines with age. And With that idea in mind, Sense Research Foundation was established, and we have the 7 strands that Michael was talking about. So I'm not going to go in detail in those, but we have funded various intramural research projects as well as extramural research projects, which are aimed to develop therapeutic interventions for those damages. With that funding, we have been successful in some degree. Like there are a lot of intramural research projects as well as some extramural research projects that have become successful biotech companies, and they're moving this approach forward. And like I was talking about the education program, we're making progress in teaching several students, and there are different at different levels, and preparing them to help solve these problems with us. My lab mainly focuses on understanding the mechanism responsible for accumulation of senescent cells and how the age affects the ability of the immune system to function normally, and the interaction between those two. Senescence has been very well studied now. We almost everybody in the audience, I'm sure, knows what cellular senescence is, at least by the classical definition. And all of this work started not really that far back. The original paper where they talked about senescence was in 1965. But it was much later where work done in my previous lab, in Judy Campisi's lab, demonstrated that there are specific phenotype in the cells that can be used to identify them in vitro and in vivo. And then later on, this whole field accelerated quite significantly, and a lot of work has happened. And now we have some understanding of what senescence looks like. But if you really dig deep into it, then we don't really quite understand a lot about senescence, and that has also impeded the field. So to briefly speaking, senescence is a state of a terminally differentiated cell when it escapes cell cycle. In most cases, that is to, at least the expectation is that it is to prevent the tumor in the body. But senescent cells can be characterized. We can study them in vivo with various markers, like we can look at the dysfunction or chromatin rearrangement, and look at senescence-associated heterochromatin foci. Like I was talking about, the cell cycle arrest markers that can be visualized in the cell. We look at the increased ROS and mitochondrial dysfunction and damage to the nuclear lamina, as well as a change in the shape and the physiology of the cell. These cells tend to accumulate lysosomal dysfunction. They accumulate a lot of damaged mitochondria, damaged lysosomes, and as a result, protein aggregates tend to accumulate in these cells. And these cells are not just sitting in isolation. They produce a complex mix of pro-inflammatory cytokines and chemokines and matrix modifying proteins, which can actually propagate the signal, or damaging signal, to neighboring cells and other cells in the body, and cause detriment. We now know that senescence is actually involved in lots of diseases and disorders. So understanding basic biology of senescence and finding ways to target them effectively can be useful in treating not only aging, but a lot of age-related diseases and disorders. However, like I was saying, there are some fundamental questions that we still don't quite understand, and that is what we are trying to do in our lab. So we don't still know how to label senescence cells, how to identify, because we don't really have a good biomarker of senescence. We also don't understand how senescence accumulate in our body. Is it because more cells tend to accumulate damage and they become senescent, or is it because the immune function declines, and with the declining immune surveillance, there is a higher buildup of senescent cells? So we don't quite understand. We also don't have a really good way of safely getting rid of them, and I'll get to that in a second. So although there are several ways we can mark senescent cells in vitro and also in vivo, really, but none of these markers, persistent DNA damage foci, lysosomal beta-galactosidase, loss of proliferation, cell cycle checkpoint marks, SASP, nuclear lamina disruption, none of these markers really are specific to senescent cells. And in many cases, in vivo, it becomes a bigger problem because there are other cells in our body that can show these markers. So combining multiple markers is one way people have used previously, but we really don't have a good marker. And that has also impeded for the development of novel senolytics, because it's hard to look at the efficacy of the drug in vivo. So we're trying to find better markers of senescence that are more useful in vivo. And in that end, we've used proteomics approach and phage display approach to identify markers. We began by looking at the surface of senescent cell, and in that process, we used this reagent, which allowed us to pull all proteins with an oxidized NCO glycan from senescent cells. And when we performed proteomics, we were confident that we're looking at proteins which have a domain that is sticking out of the cell and not something just which is present bound to just the membrane on the intracellular side. When we did that, we found various proteins that were abundant on the surface of senescent cell. We've characterized several of these proteins. I'm giving some examples here. This one protein, which we're calling SRF1, was significantly elevated on senescent cell that was treated with a chemotherapy agent, doxorubicin, which is a topoisomerase 2 inhibitor or a replicative senescent cell. And we found SRF1 was quite significantly elevated on the surface of senescent cell. Similarly, we found another protein, SRF3, which was also quite significantly elevated on the surface of senescent cell, whether it was treated with doxorubicin or replicative senescent cell. And we've been able to confirm these markers in other cell types. So we understand that senescence is very different depending on the type of the cell and the type of the damage. And by measuring the expression of this protein in multiple models of senescence induction, we were quite confidently expect SRF1 and SRF3 could be a reliable marker of senescence. We also confirmed that both these proteins were not expressed in long-term quiescent cells, but only in senescent cell. And we looked at, we've done more work with this, and we've confirmed the expression increases over a period of time. For instance, SRF1 and SRF3 are only elevated day 5 after doxorubicin treatment and plateaus after day 10 and remains high on the surface of the cell. We are currently testing these proteins in vitro in tissue sections to confirm that these can be used in vivo as well as a biomarker. But we found something very interesting when we were looking at these proteins, and I'll get to that in a second as well. The other approach we use to identify proteins or ways to tag senescent cells in vivo was slightly different. So unlike proteomics approach, which has limits of how much can we detect, how much we can go into the unknown, phage display analysis has an advantage over proteomics, because instead of looking at proteins, we are looking at or specific proteins, we're looking at motifs that are enriched on the cell of interest. So briefly, what we did here was we relied on the ability of a bacteriophage, which can bind to the RGD domain, the most common domain of all of the proteins. And then we modified that domain by using different amino acid sequences, which would either increase or decrease the binding affinity of the RGD domain. And with that, we cloned, our collaborators cloned 100 million different combinations of peptides, which were about 8 to 12 amino acids long. And each phage represents an individual peptide. And we induced senescence in endothelial cells derived from 2 different donors, treated them with doxorubicin, and then we incubated them with these bacteriophages. And we performed an affinity screen and removed all of the lightly bound proteins. And after doing a couple of cycles of such enrichment, we were able to identify some motifs that were enriched. We synthesized those peptide sequences, tagged them with a fluorophore, and then we repeated our screen, and we found several proteins which were enriched. And this is a simple heat map where blue means that there was no binding, and red means a strong binding. And we were able to hone 2 different peptides, peptide 6 and peptide 18, that we found were interesting. Meaning they were able to show strong binding on senescent cell, but we found weak binding on non-senescent cell. We've confirmed these experiments later on in other models of senescence induction, in other cell types, in humans, and as well as in mouse. And we found peptide 6, for instance, is showing a strong binding in senescent cell derived from endothelial cells from 2 different donors here. We showed that this was also showing strong binding in senescent cells derived, human IMR90 cells that were made senescent either by doxorubicin or by replicative senescent cell. We've confirmed it, like I said, in hepatocytated cells and mouse embryonic fibroblasts, so it's not limited to the species. And in this, we were able to show that in old liver tissue sample, at least, we were able to see this peptide 6 was able to co-localize with phalloidin, which stains the nuclear lamina, or sorry, not the nuclear, sorry, the actin filament in the cell. We're doing more work with this now. We're trying to identify what peptide 6 binds to. We have some ideas, we're pursuing them. And we're also confirming peptide 6 binding in other tissues. And the idea here is then to potentially use peptide 6 to track senescent cells in vivo eventually. Shifting gears a little bit, how do senescent cells accumulate with age? This has been a proverbial question that has been very of great interest to me, whether there is a decline in immune surveillance or increase in senescence burden or propagation of senescence in vivo, and we decided to tackle both of those questions. So what changes are occurring in our immune surveillance with increasing age? We focused on natural killer cells here because those are the most common, most well understood immune modulators of senescent cells. They have a very well organized mechanism to identify senescent cells based on the presence of activating or inhibiting receptors, meaning the density of those activating or inhibiting receptors. NK cell makes a choice to either kill a senescent cell or not by releasing of these proteins called perfins and granzymes, which can cause apoptosis in the target. In our study in both peripheral NK cells derived from human subjects, we found there was a decline in the number of CD56 bright CD3 negative NK cells, which are mainly NK cells that are present in peripheral circulation. We also found a similar decrease in the number of NK cells that were present in the spleen of mice. And the ability of NK cells to proliferate also significantly declines. We are planning to do some in vivo experiments where we would transplant NK cells derived from young and old mice to look how their cytotoxic function declines in vivo. But we did find that even in the burst of circumstances, NK cells can kill senescent cells more effectively than non-senescent cells, but it is not perfect. This was done in human subjects. We found that regardless of the age, in this case, so I'm showing the data from all young males and females. M is male and F is female, and this is the age of those individuals. We did not find enough, even under best enrichment conditions, we isolated these NK cells from the patients. We cultured them in vitro, and then we co-cultured them with senescent cells, and we looked at cytotoxic function that was still not as good as we would have liked. So we're finding ways to improve it. And there's several things we're doing and trying with the intention of developing immune-based therapeutics. So based on our proteomics, so we thought, okay, so NK cells has the ability to kill senescent cells, but it is not perfect. Can we improve it? And to be able to improve it, we went back to our proteomics and we found another protein called SRF5, which we think can be a biomarker of senescence as well. And we found SRF5 was significantly elevated on senescent cells. And this is by flow analysis, we compared the expression of SRF5 in IMR90 cells that was made senescence by treatment with doxorubicin. And we found this was significantly present, elevated on the surface of the cell. We've confirmed this expression of SRF5 in replicative senescent cells, adipocytated senescent cells, and also mouse tissue. And we were able to see this was elevated. We've also confirmed the expression of this protein in other tissue. And if you look at the literature, that protein is actually quite significantly elevated in several tissue across, in our body. It is highly expressed in cancer cells as well. It is generally present within the organelle, but is highly elevated on the surface of senescent cell. So we designed CAR-NK that Michael was talking about. CAR is essentially a TCR where we incorporate the SCFA, the antibody antigen recognition domain of an antibody into a construct, and we utilize the hinge of a CD28 and transmembrane domain of a CD28, and the intracellular signaling is through CD3. The idea here is that if the CAR recognize the target, in this case SRF5, we are giving another way to NK cell to kill senescent cell. So we've designed different CARs based on different antibodies that recognize different domains of this protein. And I'm showing data with one of those constructs. Here we are showing that NK92 cell, which is an NK cell line. We see that there is a killing of senescent cell when we just co-culture them with senescent cell, but that killing is enhanced even more if we included a CAR that recognizes SRF5. We are testing other CAR constructs that we've made, and once we have a good handle on the most effective CAR that we were able to test in vitro, we will then test this in vivo and as a proof of principle show whether CAR SRF5 CAR NK92 cell could be effective in finding and killing senescent cell. So like Michael was saying, my main focus was on innate immune regulation of senescent cell and what other immune cells can find and kill senescent cell. In that process, in that work that happened in the lab, we were looking at other immune cells that can find and kill senescent cell. And our focus went to this unique T cell, which is not really an adaptive T cell type, not quite really. So T cells, if we look at T cells, they are mostly classified based on the TCR composition. And most common T cells are adaptive T cells, which have an alpha and beta chain that makes it TCR. And TCR quickly is the main receptor that T cell uses to recognize the antigen, which is presented by an antigen presenting cell. So this is a basic immunology or basic adaptive immune response. The TCR changes quite significantly. That variability in TCR is what allows the immune system to adapt to different challenges and learn from it. However, there are other types of T cells which are more innate-like in function. We know about the T cells like that are present in the mucous membrane, whether in the germline or other mucous membranes. And they are also invariant-like T cells, which are present mostly in circulation. The T cell that I was going to be talking about now are called gamma delta T cells. They make very, very small percentage of circulating T cells, about 0.5 to 5%. There is variability in the population. And like I was talking about, the proportion is very, very small. So when we analyze the PBMC of human subjects, these are different humans from different age groups over here. We found the proportion of gamma delta T cell quite significantly low. But using a combination of cytokines and drugs, we were able to amplify and enrich this population by growing them in 10 days. And we were able to amplify them to about 89.4%. So starting from less than 14%, we were able to enrich this population to about 89.4%. And when we co-cultured this population of gamma delta T cells with senescent cells, we found they were very effective. So if you remember, we showed NK cells can recognize senescent cells and kill them, but the cytotoxicity was about 40 to 50% at 1-to-1 target effective ratio. But gamma delta T cells seems to be very effective. They're almost as effective in killing senescent cells, even at half of the number of targets in there. So less than half of gamma delta T cells kill as many senescent cells as NK cells were. And at 1-to-1, we were able to effectively remove all senescent cells. Since then, we have confirmed this in multiple cell types, multiple modes of senescence induction from multiple donors, and they seem to be very, very effective in finding and killing senescent cells. And one additional advantage of gamma delta T cells is that they are allogenic safe, meaning that gamma delta T cells can be, in theory at least, isolated from one individual and then enriched and then can be transplanted in another individual with limited host to graft rejection. And then we were able to show that what mechanism is involved in by which these gamma delta T cells are recognizing the target and killing them. So here we're showing that if they don't not only use NKG2D receptors, but they also use gamma delta TCR, and by blocking both those receptors, we were able to cause appreciable decline in cytotoxic function.
Amit, we're on stage with you. We invaded your stage.
Okay.
Maria and Michael, I leave it up to you. You have 5 minutes for questions, but you can finish your presentation if you prefer that. And Michael was trying to address some of the questions in the chat. Do you want to finish and we get out of here, or you want to go ahead and
let me finish my my presentation. I'll try and be quick. Yeah.
And everyone here, you can find Amit if you want to ask him directly around the room. We're gonna have a 30-minute break coming up, so you can do that. Go ahead, Amit. We'll get out of the stage.
Thank you, Maria. So we're very excited about both these projects, CAR-NK project and gamma delta T cell project, and we are testing currently right now these in looking at immune surveillance of gamma delta T cells in vitro and in vivo. If you're interested in our approach moving forward, we have Gabriel from my lab presenting a poster where he's the one who's mainly responsible in developing that in vivo experiments. He has some interesting results from mice as well. While we were looking at the proteomics of senescent cells, we found some interesting vulnerabilities, novel ways to find and kill them. I'll quickly go through some of those. Another mechanism, so the reason why we got interested in this is that another way by which senescence could be propagating in vivo is by parakine mechanism. So this paper demonstrated for the first time that actually parakine senescence could be important. Here they're showing that if you transplant the luciferase positive senescent cells in mice, then over a period of time, you can see senescent cells that are luciferase negative in different parts of the mice, suggesting that these cells, the factors these cells are producing can drive inflammation down the road. And in this context, we've looked into that actually parakine senescence is actually a mechanism by which senescence propagates in vitro as well. We've done some work on there. We know, for instance, that parakine senescent cells are actually phenotypically quite different than primary senescent cells. We've published this paper, and in this paper, we showed that not only parakine senescent cells are resistant to conventional senolytic drugs, we were able to show that they were actually vulnerable but they have another vulnerability, meaning ferroptosis. They're also resistant to ferroptosis. So in addition to being resistant to apoptosis, they were also resistant to ferroptosis. And we were able to show that by targeting them, and I'm sorry, I'm rushing things now. We can target them by using a ferroptosis inducer or a drug which can target them based on the presence of labile iron. So we cage a toxin with with a molecule that protects that toxin, and the toxin releases labile iron using Fenton reaction in cells which have high levels of it, and then eventually kills them. And we think it is pretty safe way to kill senescent cell. Published this paper recently. You can read more about it if you like. We're moving forward by testing this drug, TRX-CBI, in mouse model and trying to identify like a novel senolytic approach here. So we have some preliminary data showing that older mice, these are 18 months old mice that were treated with the TRX-CBI. So here we're showing the senescence burden by luciferase, P16-driven luciferase expression seems to be quite high. When we treat them with CBI TRX, that burden significantly declines, and GCV was the control here. Like I was saying, we found other druggable vulnerabilities by identifying these proteins. We found a novel way, which is apoptosis independent. And this drug is another drug we're testing in-house right now as a novel senolytic drug, which doesn't kill senescent cells by targeting apoptosis, but another death-resistant mechanism that they work with. And that is currently being tested in vivo. We will have those results coming up pretty soon. And this is quickly about a cytotoxic peptide that we've identified. So in addition to peptide 6, we found another peptide, peptide 18, which when co-cultured with senescent cells can kill them. With that, I'll thank the collaborators and the donors who've given us the money to do this work, and other funding sources, and the team. You'll find all these people hanging out. I'm sorry I had to rush my presentation, but I'll answer any questions you have. Thank you.
That was wonderful, Amit. Thank you. Big applause. I'm sure everyone is applauding at home, but I can't hear you. Oh, I see your reactions there, hearts and likes. And okay, awesome. Thank you, Amit. Yes, we're going to take a 30-minute break, and Amit is going to be around if you want to ask him questions. And you can always email him. Amit, you can put your email address on the chat for everyone. That'd be awesome. And we will be back at 11. You can roam around, float around, and visit our booths, visit the student booths, and network and have fun. We'll see you soon.
Yeah, and thanks, Maria. I'm going to be hanging out, and if people have any questions, I'd be happy to answer any questions anybody has. Thank you.
Fantastic. Thank you. Thanks, everyone.
Hi, everyone. My name is Lily Fishman, and I'm the Director of Academic Affairs here at SENS Research Foundation. I just wanted to come on here to thank all of our students for their fantastic work, both this summer and during the school year, and encourage everyone to stop by their posters. Students are rotating throughout both the morning, the early morning and the mid-morning sessions. So if you go see a poster that you are interested in learning some more about, please feel free to check around you, and the students should be nearby. Please feel free to click on them and start a conversation about their work. I know that they're very excited to share all of their findings from this summer and this year. So thank you very much, and I'll see you guys around. Hi, everyone. I'm pleased to introduce our first student presenter, Sahiba Dogra. Sahiba is a senior at St. Mary's College of California, where she is majoring in biology and minoring in data science. Sahiba is passionate about genetics, immunology, and neuroscience, and her prior experience is focused on using cross-disciplinary approaches to solve big, complex diseases and pathologies ranging from plant virology to respiratory pathogens. That very cross-disciplinary interest was one of the characteristics that made her perfect for her summer placement here at SRF under the advisement of Dr. Amutha Bhumanathan. Sahiba will be speaking today on euclidating the role of ATP-IF1 in mitophagy and mitochondrial biogenesis. Thank you for your time, Sahiba, and I can't wait to hear about your work that you did this summer.
Hello, everyone. Thank you, Lily, for that really sweet introduction. My name is Sahiba, and I was a summer intern at Sense Research Foundation, working with the MitoSense team, and I'm thrilled to be talking about my research, which is focusing on elucidating the role of ATP-IF1 in mitophagy and mitochondrial biogenesis. So mitochondria are the powerhouse of the cell, right? But how often have we given credit to the millions of generators, rotors, and engines working within that powerhouse, working to give us energy? And most important, what if these generators stopped working? Over 1 in 5,000 people in the US suffer from mitochondrial diseases without any cure, and many more are affected by mitochondrial dysfunction in age-related diseases like Alzheimer's and Parkinson's. Dysfunctional mitochondria are heteroplasmic, meaning that they contain a mixture of mutated mitochondrial DNA, or mtDNA, and normal mtDNA. As the level of heteroplasmy increases beyond a certain threshold, disease phenotypes become present. Therefore, by selectively eliminating mutated mtDNA, it offers a promising approach to therapeutics for mitochondrial disease. Our lab in particular seeks to use the cell's mitochondrial destruction system, also known as mitophagy, to selectively eliminate mutated mtDNA and improve mitochondrial function. So mitophagy can be induced by changes in the inner mitochondrial membrane, or IMM potential, which is made up of a gradient of hydrogen ions. The gradient is regulated by a protein complex in the IMM called ATPase complex 5. This protein complex has a synthase and hydrolase component. Its synthase component makes ATP by using the passive flow of hydrogen ions down their concentration gradient. The hydrolase component is vital to maintaining the IMM potential by pumping these hydrogen ions against their gradient. So without this hydrolase component, a stressed or dysfunctional mitochondria would not be able to stabilize itself and would undergo mitophagy. However, healthy mitochondria would still be able to function without the stabilizing properties because they would rarely be destabilized in the first place. So the protein we're looking at, ATP-IF1, inhibits this hydrolase component of ATPase complex 5. So first, that ATP-IF1 gene is expressed, which increases the production of the ATP-IF1 protein, also known as IF1 for short. Once it's produced, it dimerizes and binds to ATP synthase, which inhibits that hydrolase function, and then the IMM potential cannot be modulated, leading to a decrease in that potential, and that further leads to PINK1 and Parkin mitophagy pathways, and finally mitophagy. In the diagram shown, the two mitochondria have been destabilized by a membrane uncoupler called CCCP. When ATP-IF1 is present, it inhibits that hydrolase function, and as you can see, all the H+ ions are stuck on the inside, leading to an imbalance and ultimately mitophagy. In an absence of ATP-IF1, that hydrolase function can stabilize the cell and mitophagy won't happen. So we hypothesize that by overexpressing ATP-IF1, we will selectively induce mitophagy in dysfunctional cells, leaving those functional mitochondria unharmed. And to investigate this hypothesis, we grew out 5 stable cell lines. So our two controls are our 143B wild-type cancer cells and BH2 heteroplasmic cells with 6 mitochondrial gene deletions, which lead to deficiencies in oxidative phosphorylation, or the machinery in which ATP is produced. So our three ATP-IF1 constructs are OE, which contains a plasmid that overexpresses the ATP-IF1 gene, S39A, which contains a dephosphorylated active version of ATP-IF1, and S39E, which contains the phosphorylated and inactivated protein of ATP-IF1. And we'll be using analysis at the genomic, RNA, and functional level. qPCR is our measure of mutated mtDNA load. RT-qPCR is our measures of the levels of ATP-IF1 in mRNA form, which we use to confirm that our plasmids are expressing the gene. And then we also had functional assays to show whether the mitochondria can actually improve their function after those dysfunctional mitochondria have been cleared. So after each successive round of mitophagy, which takes about 2 weeks, we hope to see that the mutated mtDNA load decreases and that our cells show improved oxidative phosphorylation functioning in the OE and S39 cell lines, again, because we've gotten rid of those dysfunctional mitochondria through mitophagy, leaving only the functional ones behind. However, this is not what we saw. So after 4 weeks on doxycycline, which activates all of the plasmids in our cells, the constructs had the same mutated mtDNA load as our diseased control. Further, in an impedance assay, the cells could not grow on galactose, which can only be metabolized in oxidative phosphorylation. And since those cells could not grow in galactose, oxidative phosphorylation is dysfunctional. And so, in an effort to troubleshoot, we referred to an article that analyzed the effect of ATP-IF1 overexpression in fruit flies. So the authors, Kendall et al., needed to use ATP-IF1 in conjunction with an MF1 inhibitor to see an effect. So as you can see in the graph below, and specifically the region that I've highlighted with a black box, the mutated mtDNA load is much higher when just expressing ATP-IF1 alone when compared to expressing ATP-IF1 overexpressed with the MFN knockout. So what is MFN1? It's a gene called mitofusin-1 that is responsible for mitochondrial fusion. Mitochondria are not static organelles. They fuse and they split apart. And if they are stressed or dysfunctional, they can fuse. And that fusion, it actually makes them more resistant to mitophagy and prevents ATP-IF1 from doing its job to get rid of those dysfunctional mitochondria. And so we obtained a fusion inhibitor called MFI8 to test its ability to induce mitophagy when expressed with ATP-IF1. We also obtained a fission inhibitor called MDV1, or diamine-related protein 1, to observe that the effects that we're seeing are related to fusion and fission only and not any other process. And finally, we have DMSO, which is our negative control. And so after treating for a certain amount of time that we found according to the literature, we used JC1 flow cytometry and qPCR to test whether we have functional recovery and reduced mitochondrial mutation loads. So after 24 hours, we saw a surprising result. And just to let you know, so the MFI ratio is showing fluorescence intensity. So mitochondria that stain red with the JC1 dye are healthy, while those that are stained green have dysfunctional IMM potentials. So the higher the MFI ratio, the healthier the mitochondria. And as you can see, the overexpressed cells in MFI8 treatment had a significant jump in that ratio just after 24 hours, and this seemed to offer a promising hope that MFI8 with the OE construct can improve mitochondrial health. However, 1 week after treatment, that effect disappeared. As you can see, the MFI ratios in all of the MFI treatments have about leveled out. And further, we also looked at our mutated mtDNA load. And again, all of the constructs have about the same levels. There's no difference that would be physiologically significant. So once again, to kind of troubleshoot, we tried to perturb our dosage and timing of MFI-8 treatments to try and enhance that effect that we saw with overexpressed after 24 hours and try to maintain that for longer to see if that could affect our mutated mtDNA load. And so we had 3 different timelines. The first was an MFI-8 treatment that was done every about 2 days for 6 hours. And then we also had MFI-8 that was treated for 24 hours and then not anymore after that. And then our DMSO control was also for 24 hours. And so as you can see in the JC1 flow cytometry, again, the constructs are about the same with not any major differences between the constructs. However, we do see something interesting in our qPCR data that shows mutated mtDNA load. The mutated mtDNA load is higher in the DMSO controls compared to all the constructs in MFI-8. And so this seems to point to the fact that MFI-8 actually works independently of ATPIF1, and it seems to drive membrane potential changes upstream of ATPIF1. So therefore, we say that ATPIF1 cannot drive mitophagy of dysfunctional mitochondria alone. Therapeutic development, therefore, needs to consider the influence of both biogenesis and mitophagy. Future possibilities also include investigating another part of the ATPIF1 pathway, particularly the PINK1 and Parkin genes and IMM potential changes. We can also analyze upstream regulators of ATPIF1 to deduce the exact role of ATPIF1 in mitophagy. For example, this could include looking at ATPIF1 kinases and regulators of mitochondrial fusion and fission. And so as we unravel these links between the mitochondrial engines, rotors, and generators, and also fusion, fission, and mitophagy, we come closer to finding a cure for mitochondrial dysfunction. And before I end, I would like to give a huge thank you to the entire MitoSense team, including Dr. Kate, Dr. Bhumanathan, Pavna, Annika, and Jonathan, and the ApoptoSense team, Dr. Tess, Dr. Yafe, and Gabriel for their help with flow cytometry, and the Sense Education Department, including Dr. Lily and Christine, for their this wonderful opportunity. Thank you.
Thank you so much, Sahiba, and for your research and for all your hard work this summer. I'm going to throw out some questions from the chat. So Michael Ray has asked you to translate this. Is there a suitable small molecule mitofusin 1 inhibitor?
That's a good question. So mitofusin 1 is actually not looked at in vivo because of other effects that it could have. So it wouldn't be a suitable therapy, but there are definitely other therapies that are being developed to hopefully, indirectly target the fusion and fission genes.
Great to hear. Are there any other questions either in the, I guess they have to be in the chat, but I'll give everyone one more second to respond because I see one more coming in from Michael Ray. So we'll give it one more second. Still shows him typing. Michael Ray also asked, is there any reason to think that this would not work as well in quiescent or post-mitotic cells?
I think it should not make a difference. However, I'm not certain about the answer, and I would have to do a little bit more research and get back to you on that. But it's definitely
you'd have to come back for another term with us then.
Yeah.
In that scenario. Oh, but I see another question coming in. Sorry, waiting for the typers out there. Michael gave you a thumbs up. we'll take this one last question and then we'll head on to our next speaker. But Gary Abramson asked, would you like to pursue a career in rejuvenation biotechnology? Which I think is a great question. What's your future plans, Sahiba?
Yeah, I'm definitely open to it. I had a great time this summer, and there's so much within this field that I would love to explore it more. And absolutely, thank you so much.
That's fantastic to hear. We were really delighted to have you with us this summer. So thank you very much. And with that, I will turn it over to Maria.
All right. Wow. That was fantastic, Sahiba. Thank you. I'm really proud of we are really proud of our students and the incredible work that Lily is doing with our education department. Our next speaker is Dr. Jean Hebert. He's one of our extramural researchers. He's a professor at the Albert Einstein College of Medicine in New York. His presentation today is Progressive Brain Tissue Replacement and How We Beat Aging. You can read his presentation summary and bio on the events website. And now let's welcome him to our stage. Enjoy the presentation.
Hi, everyone. Thanks so much for having me. I'd like to acknowledge the SENS Research Foundation in several ways. One, for their support of our projects aimed at reversing brain aging, but also for the excellent students that they've sent our way. Recently we've had Sanjana, who's presenting a poster tomorrow, and so really excellent students. So thanks for having that program and letting us participate in it. So there are a couple of things that we work on in the lab. One is a microglia replacement project, and I think I've talked about that in the past. So I'm going to focus a little bit more on a brain tissue replacement approach and why I think that that's really important for defeating not just brain aging, but overall aging. And I'll give you my reasons why, and then I'll be happy to discuss and take questions for, if anybody else has any ideas or especially critiques. So definitely open to critiques and then being able to make this a better project. So the reason that we're thinking at the tissue level of replacing brain tissue is that really the damage that accumulates with age occurs largely outside of cells. So a lot of the focus on cellular processes with the epigenome, with mitochondria, with genetic reprogramming, et cetera, might make the cells perform better and the organs overall do better. But the bulk of aging that's occurring outside of cells is complex, it's stochastic, and it's not enzymatically driven, and it cannot be enzymatically fixed. So there are no genes encoded in our genomes that can address this damage. So examples are shown here on this slide, just for proteins. There's also carbohydrates and DNA that accumulate complex damage. And so there's no way for at least the extracellular protein damage that the cell can repair that. And without that environment, the cell behaves as an old cell, even if it's young. So in heterochronic transplants, where you take young cells, put them in an old brain, they behave like old cells, and vice versa. If you take old cells out of the brain and leave them in culture, neurons, for example, they can outlive the age of the organism. Or if you put them in a young environment, then they'll do better as well. So I think it's really important to think of, if we want to beat aging, to do that at a tissue level, tissue or greater level. So there are other people working on replacing, other parts of the body, either as a whole or progressively with different parts as you need them. But in any case, those are not what my lab works on. But in conjunction with those, I think there's a lot of promise here for actually beating aging. So is brain tissue replacement possible without a discontinuity of self or function, right? Because that's important. The whole point is to preserve who we are and continue enjoying life. So I think the answer is yes. And I'll give you the reasons for that. We're focused initially on the neocortex, which is the outer part of the brain. It's the biggest part. It's arguably the most important. It encodes our long-term memories, cognition, highest cognitive functions, consciousness. So, if we can't fix that, then, what's the purpose of working on anything else, really? So the two principles that support that progressive neocortical tissue replacement is possible is one, plasticity. So memories and functions can seamlessly move from one part of the neocortex to another. And I've given this example before, but it's only one of many examples where language, something that's very dear to us as a function, can move from one area to another if that initial area is slowly destroyed over time from a pinpoint out, in the case of a benign brain tumor, for example. So these individuals, eventually have the tumor removed, they have a big hole in their brain, but they never lost the ability to speak. And no one ever noticed any difference. They didn't notice a difference. So they're perfectly fine. And then when they go look for where speech is encoded, it's somewhere else in the neocortex. Not always the same place, but it has moved. So this is a level of plasticity that tells us that we can replace tissue in the neocortex without losing continuity of self. The other sort of established principle that supports progressive brain tissue replacement as a means of beating aging is that young neocortical precursors, when you transplant them into the adult environment, don't seem to care that they're in an adult environment and follow their innate developmental program and do remarkably well, even in the adult brain, to wire correctly, even to distant targets in the brain. So this is something that's been reproduced by many labs. We've done some work with this as well, both with mouse into mouse grafts, but also human into mouse grafts. So here's a human example where we have the human graft in green here, and it projects its axons in the correct orientation down to this major tract here, the corpus callosum, which you can see is bright green, and that's because of all the projections coming from this new brain tissue going along these tracks. And if we look further at more distant targets in the brain, we see that they're, those neurons are innervating those areas. And I'll give you a little bit more detail on that in a bit. But this is just to say that these cells, these precursors of the brain, have this established potential to generate the connections potentially that we want in the brain. And so, I mentioned other groups have done this. These are examples of high-profile papers. In this case, where they all use human precursor cells, neocortical precursor cells that they transplanted into adult rodents. And all showing all these pretty amazing properties, and many of these are from different labs, so highly reproducible. So the tumor example I gave with the plasticity, if done right, means that we can remove old tissue, and the example I gave you and these other examples from other labs show that you can add potentially new tissue. Certainly the cells seem to do very well in the adult if they're immature precursor cells. So we should be able to add new tissue that can encode information that's useful to the host and allow function to transfer, allow self-identity to transfer without us even noticing. But not there yet. So despite those very nice examples where connectivity of transplanted cells is remarkably good, it's not good enough. There are things missing. One thing that's missing is these grafts typically don't contain the full complement of cell types that are normally found in the neocortex. And all the cell types are there for a reason. They're all doing something important for function. And without them, the tissue can't function normally. So that's one thing, is they're missing cell types. Another thing is the organization of the cells is very abnormal. So if you look at, one of these acclaimed successes of transplanting organoids into the brain, you can see that the tissue is very different than what you have in a normal tissue, where you have this very nice layered structure. That is not recapitulated. Maybe in micro locations it is, but that's not good enough to get normal function, because the normal lamination is important to get normal wiring within the graft. So that's two things that are missing. So our goal is to address these missing aspects of grafts so far, to make grafts that will develop into a normal structure and will have the full complement of cells. And to do that, we don't want to reinvent the wheel. We're basing our approach on how our neocortex normally developed from a relatively simple fetal tissue at around gestational week 6, where there's a limited number of precursor cell types in a very well-defined organization that we want to recapitulate in a prototissue that we will then use to graft. And then with the anticipation that this tissue will develop the correct layers, and as already shown for disorganized tissue, will project normally outside of the graft as well. So this is an example of a couple of key aspects that have been missing from all grafts so far. In green here, there's a small number, but a very important pioneer neurons called Cajal retius neurons. They're very important for setting up the architecture and the wiring of the neocortex, the layers that come afterwards. Also very important are a stable radioglial fiber attachment to an overlying layer here that's not shown, but I'll come back to that. So these fibers guide new neurons. So these radioglial fibers are part of the radioglial stem cells that are not only stem cells, but by virtue of these fibers act as scaffold for the new neurons to migrate and form these layers in the neocortex. So here's sort of where we're at with this project. This is again the prototype tissue the proto-tissue that we want to make. It has 3 layers, this pia layer, this marginal zone, ventricular zone, doesn't matter too much for your purposes what they're called, but they have relatively few numbers of precursor cells in them. And all these cells can be derived and have been derived by us or our collaborators from human iPS cells, except for one we're still working on. And then we can also purify all these cells, again, except for the one that we haven't managed to derive from iPS cells yet. Sorry about the noise, if you can hear it. And so it's not just the cells that are important here, it's also the extracellular environment that are in to make sure that these cells kick off the right developmental program from the start. So we're doing proteomics to identify the extracellular components that surround these cells in each of the different layers. So this is an example of the transcriptomic, single-cell transcriptomic analysis that we do to compare our iPS-derived neocortical precursor cells. This is for the principal neuronal lineage, and we compare that to real human fetal neocortical tissue and find that we get a very good match. And we do this for all the cell types, again, just as sort of a quality control. And so we've also established a platform, which we've published now, showing that we can get, the major features that we want in testing these proto-tissues in the mouse, in the adult mouse. So we can create these very reproducible lesions that we can fill with our precursor cells and biogel. And then we find that all the cell types that we care about survive in this graft. And the neurons integrate. They send projections along normal axonal pathways like the corpus callosum, the major fiber tract that connects the two hemispheres, down to subcortical targets like the striatum. More importantly, they also seem to mature normally. Initially, there's very little activity, but over time, these human neurons start to show action potentials as they normally would. And moreover, if we put the graft in the visual cortex, over time, the neurons will respond to light. So this, hopefully you can see this little orange arrowhead here is the onset of the light stimulus for these mice. And you can see that the neurons in the graft initially after 4 weeks are weakly responsive, but then become more robustly responsive over time. We also, very importantly, have vascularization of the graft. So we include vascular endothelial cells in our grafts. Here they're labeled with GFP, so we can see them in green. They rapidly form vessels. Those vessels fuse with invading host vessels. And you can see an example of this anastomosis here. And if we put a circulating dye in the animal that goes throughout the whole system, we can see that that dye goes through these green vessels as well, not shown here, but I may have shown that previously. So we have this platform that's pretty good. We can also layer cells in this platform. Again, these are not the layers that we want yet. They're just a proof of concept that if we use a biogel that's commercially available, very popular one that supports neuronal survival, we can layer green GFP labeled cells and then overlay, I think in this case it's TD tomato labeled cells, and this border is maintained, but blood vessels can still cross, as do neuronal projections, which is the properties that we would want. So we have this platform now that we can test with tissues that are more and more resembling the normal fetal tissue. But for that, we have a little ways to go. We can isolate pretty much all the cells, as I mentioned, -1. We've made quite a bit of progress on identifying key components in the extracellular matrix to make the biogels that these cells are going to initially be embedded in. And then, of course, there's going to be, a fairly extensive phase of testing whether the tissue develops into these mature layers, neuronal layers, and whether it can encode function. So we have outlined experiments that will prove that the neuronal function of the grafts, their electrophysiological activity, is encoding useful information to the host. And then if we can do that, then, success. But we might need to do a few iterations before we can do that. So that would fall into the bigger project here, a bigger phase that comes afterwards, where we're not only adding new tissue, but we have to remove the old tissue. So we're going to want to mimic what I showed you as an example with the benign glioma that grows slowly from a pinpoint out and destroys different functional areas. Of course, we don't want to use gliomas, because when benign, there's a risk that they will become glioblastomas, and that's definitely not worth the risk. So there are other ways that we can do this that we've designed on paper, but these have to be developed in reality, where we use an optogenetic approach to start silencing areas from a pinpoint out. And as long as the individual is using those functions, they will get re-encoded elsewhere, and they won't notice a difference. And then once that tissue is electrophysiologically dead, we can remove it. So that's shown here with the silencing of this tissue. So this is our prototype tissue that we add in. The brain shrinks with age, so we can add some tissue without removing anything initially. But then we have to remove the old tissue, and that's what's shown here with the method I just described. And then once that's removed, we can add the new prototype tissue. It will also grow in, and we repeat this. And in a few iterations, we have a completely young neocortex again. And this would be combined if we wanted to really beat brain aging, which we really do want to do, but don't have the resources to be working on these other parts of the brain yet. We would want to do this in combination with other parts of the brain, particularly the ones outlined here. So we need more people working on this, basically. And we already have, a lot of collaborators and a lot of people in the lab who have worked on this. I don't know if Sanjana is in the audience, but apologies, I didn't update this acknowledgement slide, but Sanjana has been a big help here as well. Yeah, and also to try to accelerate things, here's a shameless plug for a company I've started where we're basically focusing on trying to develop this tissue initially to treat patients, but it would be applicable to addressing aging, and combined with replacement of the rest of the body. I think that we can, in the foreseeable future, actually beat aging, not just live healthier lives as we nevertheless age, but actually beat aging and then decide, when we've had enough, or keep going as long as we want. Okay, so let's see. Guess I can stop sharing, and hopefully I left some time for questions.
Okay, Dr. Hebert, fantastic, exciting, and provocative as ever. Thank you very much. I am so delighted we started funding your work. Unsurprisingly, we have a whole bunch of questions. So the first one comes from Dr. Sharma, who's an immunologist, and therefore he asks an immunological question. He says, the old brain tends to have senescent cells, such as in the glia, and also infiltration of T cells. How do you think their presence might affect the engraftment of the new tissue?
Yeah, totally a valid question. We have to go with what we know so far, which is even with naked cells transplanted in humans to treat Parkinson's, for example. So, back in the 80s in Sweden, they started using dissociated fetal cells from midbrain to treat Parkinson's. And those, in post-mortem analyses, showed no signs of no pathogenic signs for at least a decade and a half. And then in one case, they had a patient that lived where they collected the brain 24 years after the transplant. And those were starting to show pathology. And we know these brains are inflamed, but those neurons were still functioning quite normally until for a long time, decades, and those were naked cells. So our expectation is that cells that are in a new tissue, so they're not just naked cells, will be first more insulated from any negative effects of the old brain environment, but also that this is over time we're going to replace all the parts of the brain so that it will be completely young. So I think our tissue will certainly do well enough for long enough for that process to occur. That's the expectation. Thanks.
I think you have a more optimistic read on those Parkinson's transplant studies than I do, but we can have that argument another day. I think he was actually asking a question that was more proximate, which is like not will the cells survive for decades once they're implanted, but when you initially are trying to implant these, what do you think is the effect of the presence of, in aging, there is already infiltration of T cells, there is already senescent cells, etc. If you're trying to do this in an aged brain, how do you suppose that's going to affect the initial engraftment in the first place?
Yeah, again, we have not done enough in mice, but maybe more has been done in humans already with these, examples of Parkinson's transplants, these are not young people that are getting these cells, and they seem to be surviving and doing quite well. There's a very recent example from Blue Rock Therapeutics, where they have these wonderful dopaminergic precursors. They're in phase 1-2 trials, and they're getting positive effects, not just for safety, but efficacy after, I guess now it's like 10 months. So it's still early stage, but I think there is reason to be optimistic, but I'm very happy to discuss potential challenges and caveats.
Yeah. Another thing I would add on just the immune side of things is, of course, that if one was working with iPS cells, that eliminates the allogeneic concern. So that actually was a question that I think addresses one of Yafei's question right there, which is if you start with iPS cells, which is actually what you're doing in your animal experiments for the most part, you avoid the allogeneicity problem. He also asks, when considering the aging brain, it's apparent that the entire organ may be affected. How can transplantation of a portion of brain tissue to enhance cognitive function? And I think this question sort of answers itself, but so let me ask it a bit more broadly than he's asked it. It's apparent that the entire organ may be affected. How are you going to handle areas outside of the neocortex where you're focusing?
Yeah, that's a very good question too. Undoubtedly, the whole brain is affected, and undoubtedly, every part of the brain is important for function. And so we need to address all parts of the brain. Again, we're going to be, I think, guided, although again, we haven't started for the other parts of the brain, but I believe we're going to be guided by normal development. And so, for example, the visual thalamus would be replaced at the same time unilaterally as the primary visual cortex, for example, so that they wire together the same way they would during development. But yes, you could replace the whole neocortex, but then you'll get a stroke into some subcortical structure, and that will affect function, or or just things will stop working. And so you don't get proper information flow from the outside world to your conscious cortex. So yes, those all have to be addressed. So this is a monster project. I hope I make it sound like it was simple. But the takeaway too is that the result is a monster result too. I mean, I think we can beat it, right? So that's, you got to weigh the pros and the cons here.
Yeah. Well, and of course you had to start somewhere. Literally, I mean, you had to start somewhere. And so, like, the neocortex is a very high value target, to say the least.
Right.
So yeah, but absolutely a perfectly intelligent question. Like, as you say, if you get a massive damage in a subcortical structure, it's not going to matter if your neocortex is functioning. I'm going to keep asking questions until someone throws me off. So, since Jason Zhao asks, since the brain will continuously atrophy over time and each surgery will replace only a small percentage of the brain, what is your best estimate of how often brain tissue replacement surgeries would need to be done in order to keep the brain healthy indefinitely?
So I think we could replace the entire brain with like a half dozen to 10 surgeries, which sounds like a lot, but again, that's not something that's not doable. And surgical techniques are getting better and better. I haven't been keeping up with surgical robots, but they're making a lot of progress, a lot faster than I thought. And that reduces complications of surgery. So there is risks with surgeries, of course, but I think those will be diminishing and a little more controllable. So yeah, 6 to 10 surgeries over the course of 20 years, and then you basically have a young brain again. I don't want to say like a teenager brain again, because that'll scare a lot of people. But you have a young brain that will last for decades, and then you don't have to do anything for a long time.
I will also add from the SENS perspective that once you've done that, you can slow down the further progression of damage by doing all the other damage repair strategies that SENS has. So once you have a nicely pristine young brain in there, it's going to be a lot easier to maintain it by doing things like removing amyloid and alpha-synuclein aggregates and direct molecular level repair of the extracellular matrix. But if you've gone and replaced it wholesale the way you're doing, that's a very, very systematic approach, to say the very least.
Yeah.
If I'm still allowed to ask you questions, Laura Lynn asks, are there any concerns about the invasiveness of multiple brain surgeries?
Yeah. So I mean, I kind of addressed that a little bit there, and I think Maria's appearance here is telling us
You can answer that question, and then we have to wrap.
It is. There are complications from brain surgeries. Not many deaths, because you can actually do away with a big part of your brain without dying. But there are complications, and those we'd want to avoid those. And so surgical techniques that are minimizing that are being progressively improved upon. And at some point, I think the risk will be low enough in a short time. I mean, these are AI-driven smart robots, and I think progress there will be that'll be ready by the time we're ready to go, I think.
Super. You've got another good question and a good comment and another question I'd like to ask you sometime, but I will get off and people can perhaps approach you privately if you're hanging around. Sure. And thank you, as always, for a great presentation, Dr. Hebert.
Yeah.
Yes.
Thank you.
Sean. Thank you for all the work you do. So you're going to stick around a little bit so people can find you?
Yeah, I'm actually traveling, but I can stick around. I'll stick around for a little bit. Yeah.
Okay, thank you. So that was our last presentation. Thank you, Michael. Thank you, Sean. And I will make an announcement in a different way so that everyone can hear me and not only the people who are on stage. So I'll be right with you on the screen in a second. Thank you. Okay, I'm back. All right. Well, I would like to take a brief moment to acknowledge our dedicated and passionate staff, founders, and directors. Their unwavering commitment and relentless efforts have brought incredible value to the field of rejuvenation biotechnology, and we are proud to say that SENS Research Foundation was instrumental in its development. I remember when we first embarked on this journey many years ago, and it felt like a wild, audacious idea to many. The concept of rejuvenating and extending human life was met with skepticism and controversy. But today it has become a recognized and respected scientific field, which was our dream and our goal since day one. As we continue to push the boundaries of scientific discovery, I am confident that the SENS Research Foundation will remain at the forefront, leading the way towards a future where aging is no longer an inevitable barrier to human potential. Last but most importantly, I want to extend my deepest gratitude to our esteemed donors on behalf of the Foundation. Your invaluable support is not just making history, it is forging a path towards a profoundly brighter future for all of humanity. Your contributions are truly monumental. Thank you so much. Okay, we hope that you enjoy the event, and we hope to see you again tomorrow. Feel free to levitate around until 1 p.m. And meet like-minded people who understand why addressing aging medically is so important. And come say hello. I'll be here. See you around and see you tomorrow. Bye.